And our last talk of the session will be Jing.
And two complementary data visualization tools from UC Santa Cruz. So I'll be the one presenting the Xena Single Cell Browser. So Xena is an online multi-omic data visualization tool. And then we can support pretty much any type of matrix data, which include the single cell RNA-seq, copy number, protein expression data, as well as cell annotations. So all the DCP data matrices are already preloaded on our portal and then ready to be visualized. We have bidirectional links to Cell Browser, which Max will talk about in the next presentation, to provide complementary views of the same data. So our main visualization is called visual spreadsheet. So there are columns and rows in a spreadsheet. Columns can be an expression from a single gene, or expression from a set of genes, or a cell annotation. And a single row is a single cell. So we have highly optimized our visualization to display 1 million cells dynamically. So what you see here is a screenshot of 800,000 cells from the Sensitive Immune Cell Project. So there are a total of 26 parameters across the entire spreadsheet. And to display that over 800,000 cells takes about 10 seconds over UC Santa Cruz campus network speed. And then we don't use any data downsampling in generating this view. So a unique feature of Xena is that Xena browser can jointly display data from multiple backends. So for example, the user have some private data, like Louvain cluster memberships, maybe hosted locally on their laptop. And the Xena browser can combine that data together with public data that's already hosted in the cloud via public hub. So what that feature will enable users to dynamically and quickly reevaluate their analysis results against a common gene expression data resource. So here I'm going to show a short video of the demo of a tool. So how do I get this started? Maybe click. Oh, so started. So here's the demo. And wait, is it? I don't. Sorry. I don't think that's actually playing. Oh, wait. Is that? Now it's playing. OK, so this is our landing page, singlecell.xenahubs.net on the top. And so here you see a live example of a spreadsheet we pre-built it. I don't think it's playing again. I think it just stuck. No, that's no. It didn't really play.
What do you want?
This is a video.
Yeah.
How do you? That's a slide. So next, this is a video. I need to start play.
Yeah, and after?
Maybe no.
Work is going.
I don't think it's playing. Okay.
It's not running. I'm sorry.
Okay. That's really unfortunate. It's going to be hard to describe it. Jesus. You're kidding me. Okay, so I'll do it verbally. Maybe this thing. Oh, wait, now it's working. Here we go. I think it's working. So we have the data preloaded, and then you can click into any one of the studies to see more detail about it. And here in there, we have a link back to the HCA data portal. And so visualize the data, you could either click this button here on the page or simply starts here. And that leads you to an on-screen wizard to build your spreadsheet. And so step one is to select the study you want to view. And you can see there's almost 800,000 cells that will be in the spreadsheet. So the first thing is let's add some cell annotation information. So let's add Lewin clusters, where the organ parts the cell comes from. And additional phenotypes are available here. So now let's add some gene expression data. And you can add any gene of your interest. So here we display the gene expression data in shade of blue to red. Red representing overexpression, and the color legend are shown at the bottom. So if you want to know the exact expression value, you are using tooltip. Here you can see the expression value of that cell right underneath the crosshair symbol. So in addition to see just a single gene expression, you can also see a list of genes expression. So here I have two lists that are pre-built. So now let's add the expression of a cluster 3 markers, as well as the expression from the markers for cluster 5 and 7. You can resize a column dynamically once it's built. You can also rearrange columns. So here, after rearranging, all the 800,000 cells will be dynamically resorted in real time. So in Xena, we always start sorting the cells first by the very first data column, column B. And ties will be further broken by subsequent columns from left to right. You can click and drag to zoom into the cell of your interest and zoom out. You can also remove the columns. So here's a different spreadsheet. Here we provide our user a complementary view of the same data by clicking that button to go to Max 2, which he will talk about in the next presentation. Okay, so thank you for here's our entire development team, and thank you for our funding region. Thanks. Sorry about the hiccup.
Thank you.
I'm also from UCSC. Thanks to the organizers for giving the portals time here, and let me speak for 180 seconds. The whole team is here, so that's me and Matt, and we get money, and we are mentored by Jim Kent. And so the aim of our little cell browser is that it shows you, it's a website, it shows you the static freeze of the data. It's mostly based on a Tisney view. Because it's a frozen view, it's your data, so we import Seurat, ScanPy, CellRanger, or any other format you have, so it's the result of the author's paper. We have a couple dozen data sets, depending on how you count, between 50 to 100. And part of those are the HCA data sets, most of the HCA data sets that are imported via Jing's Xena. And you can also run this yourself on your own data and put the result on your own web server. So it doesn't require a server, something, it's just HTML files. Because this is a demo, you go to cells, ucsc.edu, and then you have a list of data sets. Oops. What's going on? Oh, hi, thank you. Okay. There's a list of data sets, and then you can open the HCA part. It's number 4 or something in the list. And then of the HCA collection, we can open, for example, the pancreas data set, and then you get this Tisney view, and you're not limited to the Tisney view. You can do others, or a UMAP, or whatever you like. And the left side of the screen is all the cell annotations, so you can color by these, like the other portals. The right side is the legend. You can click into that to select the cells. You can also use selection tools. Oops. And then you get an overview of what you just selected. You can also color by genes, like the other portals. And you can color by two different things, like one gene and another gene. And there's a little bit of a heat map viewer at the bottom. That's mostly it. And I was thinking yesterday in the talks, when you talk about the portal idea, that by project, that Lung, that maybe I should be building, can I get back the slides? Maybe I should be building different URLs, like because I already have data collections, organoid atlas and kidney atlas, and maybe I should be building lung atlas.cells.ucs.edu with all the data sets of a certain project here. So they're all together, even if they're not yet in the HCA portal, or they're from separate groups or something. So they would be all together, and you can flip between them and color by things and get over summary statistics. So this is just a summary of the other portals and mine. And yeah, I'm happy to take questions for 30 seconds. Thanks.